Greetings, students. This is Mr. Labonte. I am going to be talking about diffusion, osmosis, and cell membranes. So the first structure that we have of a cell is going to be the membrane, and we're going to be talking about how particles move through that membrane. So the first thing we need to do is we need to start with particle movement in general and move from there. In class, I'll be going over both of these demos, so you get a sense of that. All right, so first of all, the concept of diffusion. So diffusion is movement of a particle from high to low concentration. And it's really it's created because of the second law of thermodynamics, entropy. So that is to say, all things move towards their most chaotic or their least energetic state. And if you start with a bunch of dye right here, in time, that dye is going to move from an area of high concentration to low concentration. So there's energy there. This is the least energetic state, or will be the least energetic state when it's fully diffuse, and the most energetic state when it's in its most concentrated form. So this is the idea of diffusion. So this is the kind of straightforward thing. You can think about it with scented candles or somebody farting in a room. You go from a high concentration to low concentration. It helps to understand a couple of terms when you're looking at that. So first of all, we've gone over these in class a little bit. You have solute. So this is the thing dissolved in water. For our purposes, it is always water, right? Solvent, and for our purposes, the solvent is always water, the thing that's doing the dissolving. And then together, what we end up having is a solution. Diffusion occurs whenever it can occur. So in this case, you see there's some kind of a membrane or something that is standing in the way of the particles. So occasionally those particles will hit the membrane and bounce back. But if those particles can move through, then eventually what you're going to end up with is the lowest energy state, or you're going to have an equal number of particles on one side as there is on the other side, and that is called equilibrium, right? So same number of particles on one side or the other side. Equilibrium is reached and can be reached even when you have multiple different particles. So for instance, you've got a bunch of purple particles here and a bunch of yellow particles here. Both of these groups are going to reach equilibrium. So that is to say that the particles are independent. They're going to want to move and reach equilibrium regardless of what's going on the other side. So now you have an equal number of purple on both sides and an equal number of yellow on both sides when both of them reach equilibrium. They may not reach equilibrium at the same time, but both of them are going to reach equilibrium independent of the other one. Now, how does this matter? What do we see this as? This is, in fact, and we'll get into the structure of this in a second, this is a cell membrane. And if a particle can move across the cell membrane, so I'm going to give you an example of a particle that can move across the cell membrane. In a little bit, I'll explain why it can move across the cell membrane. So this is O2. This is an oxygen particle. And let's just say I have 50 O2 on this side, and I have 10 O2 on this side. Well, if that's the case, what we're going to have is we are going to need to reach equilibrium, and we're going to end up with 30 O2 on this side and 30 O2 on this side, right? So that's equilibrium. You're going to end up with an equal number on each side. So you'll have a net movement of 20 O2 in this direction. Now it's important to note that as 20 O2 are going over here, you are occasionally going to have particles go back that way. And so you'll have oxygens moving in both directions at the same time. And that's called dynamic equilibrium, which is once you get to 30, you'll end up with dynamic equilibrium. So you will have particles continuing to move from one side to the other. It never becomes static in a case like this, but you just end up with roughly, I guess I should put a little, roughly 30 oxygen molecules on each side. So that's dynamic. Okay, so this is what a cell membrane, quote unquote, actually looks like. Note that this is the extracellular matrix. Extra means outside. So that's the outside of the cell, and then this is the internal. Cellular matrix. I will also call this the cytosol. So you have an outside and an inside. And hopefully you can see these little phospholipids. So we're going to talk about the structure of those in a second. But you still have this exact same thing, and there's just a bunch of other things being added. These are proteins. You can see that here's, it looks like a glycolipid, but it's mostly a carbohydrate that's sitting in there. That's a cholesterol molecule. So there's a bunch of different things that make up a cell membrane. But generally what you have, the most important part is this lipid bilayer. And so we're going to look at that fairly closely. The lipid bilayer is made up of these, it's a phospholipid bilayer. I'll sometimes just call it the lipid bilayer. Phospholipid bilayer is made up of these things called phospholipids. So hopefully you can see something you recognize here. You have a saturated and an unsaturated fatty acid. So this is a diglyceride. Why is it a glyceride? You can see the glycerol molecule right there, right? So you have a diglyceride, that's like sticking out your two fingers. And then, if you remember this from the nucleic acids, you have the phosphate group. So putting all that together, it's called a phospholipid, okay? And what's super important that you understand from this is that you end up with one part of it, which is non-polar. Remember, there's no oxygen here. There's just carbon and hydrogen the whole way around. And you end up with another part, this part, which is polar. Why is it polar? Well, if you look right here, you have a negative. You also have a positive right here. So you have a lot of charged particles there. Well, why does it matter if we have a nonpolar and a polar molecule sitting together in the same spot? I'm so glad you asked. Get my eraser to work. If you recall, you are made of water, right? So if you're made of water, we can assume that there is water over here on this side and water over here on this side. And what I want you to recognize is the polar part is going to be attracted to water there. The polar part is going to be attracted to water here. And then this non-polar part, or the hydrophobic part, they're going to join together to avoid water, right? So this molecule, which is this collection of molecules, is going to make up the cell membrane. It does not take DNA. It does not take planning. Nobody has to sit around telling these molecules where to go, simply because they are, remember, amphiphilic, they're amphiphilic with a polar head and a nonpolar tail. When you put them in water and shake them up a little bit, you are going to get a lipid remember bi is 2, layer. So 2 layers of lipid, or a phospholipid bilayer. 2 layers of phospholipids in this case. This is just another image of the exact same thing. Hydrophilic, water-loving head, so this is charged. Hydrophobic, water-hating tail, non-polar. This is in fact what they look like. You can see that there is an unsaturated, right? Unsaturated bond there, and you have no water in here, and then water over here on the outside being attracted to those polar components. And this is a cell membrane. So that's what a cell membrane looks like. It is a phospholipid bilayer. Phospholipid, how many layers? 2. Remember, that's 1 layer, and that's the next layer. Too far? All right. So this image is showing you that while it may at first blush help you to think of a phospholipid bilayer like a balloon or something, so like a thin membrane that distinguishes between the inside and the outside, it really is not the best analogy for a cell membrane. The best analogy for a cell membrane that I have been able to come up with would be, imagine you have a pool, and then imagine you have, like, I don't know, 500,000 of those little plastic balls that you put in like a jumpy house, and you just dumped them all on top of the pool. And so these phospholipids are kind of like the little balls in the jumpy house. And so that they would layer all the way on top of the pool, and they would jam right next to each other, so much so maybe that you would have a double layer of these phospholipids. And the reason that is a better analogy is because the orientation of each of these, so let's call this one A and this one's B, is independent. 5 minutes later, or even 200ths of a second later, B and A can switch places. So this is happening all of the time. You have all these little balls floating around in the pool, and they're all switching places. Even they'll switch sides, so they'll switch orientations. About 1 time a month, you'll have whatever, let's call this one number 1 and number 2. Number 1 and number 2 will switch places and flip over. And so you can actually have movement both laterally and vertically in the phospholipid bilayer. So it's quite dynamic. All right. How dynamic it is depends on how saturated your fatty acids are. And so this, it says hydrocarbon. We've discussed this as fatty acids. But hydrocarbons shouldn't be just a bunch of hydrogen and carbons. That's all hydrocarbon means. So fatty acids. So you should see that this spot right there, that would be the unsaturated spot. And this one, since there's no bends, then that's the saturated spot. Something that might be kind of interesting to some of you. Remember that heat is a measure of motion. So a question that I might have is, what would it take for these to be at the same level of fluidity or the same level of motion? And the answer is that this would have to be in a hotter environment, right? Because it's more closely packed together, they're not going to move as much. And so the truth of the matter is, fish that live in cold environments, because there's less motion, their cell membranes look like this, and fish that live in hot environments, their cell membranes look more like that. And so you may have heard it's better to eat fish from cold environments. And the reason it's better to eat fish from cold environments is you are eating more unsaturated fats. There's more unsaturated fats from fish in cold environments than in warm environments. There's another, so that this is this idea that fluidity is determined by the number of double bonds in the fatty acids. It is also determined by the amount of cholesterol. So we were introduced to cholesterol in the last couple of classes as a kind of a bad thing, but it's also a super essential molecule in your body. All of your hormones come from cholesterol. Most of your neurotransmitters come from cholesterol. So it's very, very important, and you can get it by well, I don't know that the neurotransmitter thing is right. I'll have to check that. But the other thing that it does is it will help stabilize this phospholipid bilayer. So as it sits in here, it helps hold the phospholipid bilayer together. All right. So how is it that molecules get across? They enter and exit the cells. So if a molecule is hydrophobic, it's simply going to move right through the phospholipid bilayer. If a molecule is hydrophilic, it's going to need a protein channel. So I'm going to show you both of these things. Here's our trusty phospholipid bilayer, and you'll remember I started with a molecule O2. So because oxygen has an equal electronegativity, this is non-polar. And you have to remember that actually there's already oxygen here. So this oxygen has been dissolved in the water or in your cytostolic fluid, or your extracellular fluids. You have a bunch of them over here. They are dissolved. They're bouncing into each other. And so because they're bouncing into each other, they can also get pushed away from each other and move into the middle of the cell membrane. So right here, remember, you have nonpolar and you have a molecule that is nonpolar. So molecules that are nonpolar can move right through the cell membrane. Another molecule that can do this, so another nonpolar molecule is, let me change my color here, is carbon dioxide. So CO2 is also nonpolar and can diffuse through the membrane. And so classically in the cells in your body, you have carbon dioxide leaving the cells and oxygen moving in simply because of diffusion, right? That is, there's more carbon dioxide on the inside of your cells and more oxygen outside. And so those two particles move in that way. However, if you had another molecule, so let's say a chlorine atom, and I'm drawing chlorine with a negative charge here because chlorine is generally found as an anion, right? In the states that we find it in. Chlorine is very, very polar. So it cannot go in through the membrane. It gets to this hydrophobic region and it is too attracted to the polar regions here. So chlorine, even though you have a concentration difference, is not going to diffuse through the membrane. Same would be true if you had something on the inside. So let's say you had sodium on the inside. Sodium, because it's charged, doesn't matter whether it's an anion or a cation, would be trapped on one side of the membrane or the other in this example. However, there are ways to get molecules that are polar across. So here we go. This one is a nonpolar, right? Here's a nonpolar small molecule. These would be polar molecules. And the way that you get them across is you use a trans membrane protein. So these proteins they go all the way across the phospholipid bilayer, and they serve like little safety guards or whatever, helping polar molecules cross and go from an area of high concentration to an area of low concentration. And this is called facilitated diffusion because you're still having diffusion occur. You're still going from a high concentration to a low concentration, but you need some kind of help or some kind of facilitation. And so facilitated diffusion occurs with transmembrane proteins. It is also sometimes the case that you can move molecules from an area, so we're going to look at these little circular ones, from an area of low concentration to an area of high concentration. So this is against the concentration gradient, right? And so in order to move, so this is these little balls that are moving, right? In order to move molecules against the concentration gradient, it requires active transport, and that requires the use of energy. So it requires energy to do active transport, and these are called transport proteins. And there's another slide that'll define the different proteins. So this is the one that we were just speaking of. So this is an active transport, and so if we're going to use this coloration, we might say that there are, we'll put a bunch of these molecules out here. And we would say that we are moving these red molecules out. All right, energy, well, it says right here, energy is required, right, to move from a low concentration to a high concentration. Channel proteins are responsible for facilitated diffusion. And so in this case, what we have is we have a bunch of molecules here. So this is high concentration, and we are going to a low concentration. So that's a channel protein. And then at the end, we have these receptor proteins, and these are kind of interesting. So some of you may have these like little doorbells in your house where when you press on the doorbell, a picture of the person who's ringing the door comes up. That's what receptor proteins are like. So no material moves across the membrane. So no movement of material. The only thing that's moving across the membrane is information. Information only. So just like the picture of the person, you know who's at the door, but you don't have to open the door. So that's what a receptor protein will do. All right, and now we're going to look at this special case of the movement of water across the cell membrane. Again, because we are aquatic organisms, the movement of water and everything that has to do with water matters a lot. And so water is going to move generally from the side with a higher solute concentration, that is, less water, so towards the side with a higher solute concentration or less water. So I'm going to draw a little diagram here. And so let's pretend I put in some kind of a membrane, and in this membrane, it's possible for water to move across the membrane. So water can move from one place to another. But let's just say I put a bunch of these little green dots in, and these green dots are really, really big. So these green dots cannot move across the membrane. But these green dots are solute, right? So they are in the solution, and they are trapped on this side. If that's the case, then what's going to happen is we are going to end up with what's called a water potential or water pressure that's higher on this side higher water potential or pressure and lower water potential, also called pressure. It is water potential, but it helps to think of it as pressure. So you're moving from higher water pressure to lower water pressure, and as a consequence, the water is going to move into the side with a higher solute concentration. So hopefully you can see there's a higher solute concentration on this side, and that's how the water is going to move. In your body, this ends up having a big impact, okay? So if you remember that a solute, or the stuff in your body that's dissolved, is going to lower the water pressure, this should help you remember it. So I'm not a huge fan of this image, but in this case, you can think of this as low solute on the outside, high solute on the inside. So high solute is going to translate into low pressure. So the water is going to enter. This arrow indicates the movement of water. In this case, you're going to have high solute on the outside, which equals low water pressure. And so the water is going to move that way. And then when it's isotonic, you have equal solute and equal pressure. So you're going to end up with this situation of what's called dynamic equilibrium. I tried to introduce that to begin with, and that's just an equal motion. These have special names. Hypotonic, isotonic, and hypertonic as far as the solutions are concerned. And this describes the relationship of the cell to solution. So hypo is below, tonic is pressure. So this is low pressure. So when the cell is at low pressure to the solution has a higher water potential or water pressure, so the water is going to go in. Hyper, like hyperactive, is high too. So this is high pressure, tonic. So when the cell is at high pressure to the solution, the water is going to leave the cell. The cell will become smaller or more shriveled, and that's called plasmolyzed. So a cell can be turgid. If it's a plant cell, that's normal, but with a human cell or animal cell, that's bad. It'll explode. Flaccid, with a plant cell, that is bad, but a human cell, that's fine. That's isotonic solution, so you have an equal amount, dynamic equilibrium. And shriveled or plasmolyzed, and in both the human and the plant cases, that is a bad scenario. All right. And then you can ask, and this is something I would do with my class when we were together, but you should ask yourselves these questions. I understand or could state why cells in a hypertonic solution will lose water, and maybe I have no idea why that's going to happen. And then finally, I thought, I'd give you an example of a case where where we can see this playing out in a situation in real life. So glucose is both large and polar, right? So we have a large and polar molecule. So glucose is not going to be able to come into your body through the cell membrane. It could try, but it's not going to do that. And so it needs to have a protein that's going to allow that to happen. And a kind of a cool thing is, so this is a receptor protein right here. So remember, this is like the doorbell. When glucose is present in the extracellular matrix, insulin will bond to the receptor protein. And when that happens, you will have these vesicles that fuse onto the membrane, and we'll talk a little bit about how that happens. And what they will do is they will put in these facilitation proteins, or these transmembrane proteins, that are going to allow facilitated diffusion to happen. And that's going to allow glucose to enter the cell. Without insulin, that can't happen. When you need to take insulin, the reason you need to take insulin is if you don't have it, you will not allow these proteins that allow glucose to move through, these facilitation proteins, to bond to the cell. So that ends up being pretty important for your body. Okay, and we'll talk a little more about that in class.